If I was to ask you a question to kind of get this session started, I'm curious as to what your answer would be. But what would you consider to be the most significant human accomplishment of all time? What's that? Okay. And then the other question would be, what would you consider to be the most significant scientific accomplishment of all time? Now, I'm sure that all of us would give very different answers to those questions. And we probably could give good reasons why we selected a particular accomplishment as being the pinnacle of human achievement. But for me, as a biochemist and as somebody who spent most of my life studying the life sciences, I think the sequencing of the human genome was the most significant scientific accomplishment and maybe the most significant human accomplishment of all time. Why? Because the human genome essentially is the genetic blueprint for human beings. And from that genetic blueprint, we can get a lot of very important fundamental information about the very nature of humanity, at least in a biological sense. And the human genome draft sequence was published in June of 2000, June 20th, 2000. And this is a picture of Bill Clinton in the Rose Garden announcing the sequence of the human genome. And on his left and right are two scientific rivals, Francis Collins on the left and Craig Venter on the right. Francis Collins headed up the Human Genome Research Program funded by the National Institutes of Health and the Wellcome Trust in Great Britain. It was a public enterprise that had been underway for almost 15 years, spending about $3.2 billion to sequence the human genome. On the right was Craig Venter, who was the president of Celera Genomics. It was a private company trying to sequence the human genome, and it was racing against the public Human Genome Research Program with the idea that if they could sequence it first, they would figure out a way to monetize the human genome sequence. And it turns out that these bitter scientific rivals decided to call a truce and agreed to walk arm in arm across the finish line together, sharing data and then producing the draft sequence of the human genome. And of course, Bill Clinton was there to announce that accomplishment. And this is what Bill Clinton said that day. Today we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, and the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all of our lives and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. So Clinton appreciated very much the significance of having the sequence of the human genome. And again, he described this as the language in which God created life. But yet, the initial analysis of the human genome sequence suggested to many people that this genome was the product of anything but the language in which God created. But rather, they looked at this and they saw what they interpreted to be vestiges of a vast evolutionary history. They argued that the human genome sequence provides us with the most compelling evidence that human beings must have evolved, and that a creator had nothing at all to do with the origin of humanity. And so what I want to discuss with you today is essentially why people arrived at that conclusion, and then I want to show you some new insights into the human genome that are radically changing the way that we think about the human genome and the genomes of other creatures. Now, a little bit of background information is in order. This is a cartoon showing the structure of DNA. And DNA consists of two molecular chains that are formed by linking together subunits into a linear fashion. And those subunits are called nucleotides, and they're abbreviated as A, G, C, and T, which are called the genetic letters. And it's the sequence of those genetic letters along the chain of the DNA molecule that contains information, that contains the genetic information to specify a human being. And DNA has a ladder-like architecture where there's actually two molecular chains that align in a parallel fashion, where the backbone of the chains are like the upright of a ladder, the side groups are like the rungs, and if you twist it, you get this well-known DNA double helix. Now, in the human genome, there are 3.2 billion genetic letters. And those genetic letters are distributed among 24 DNA molecules. And those DNA molecules are packaged into structures called chromosomes, where you have the DNA double helix wrapping around this protein complex called a histone core. And the DNA double helix will wrap 1.5 times around the histone core. There's a linker piece of DNA. It'll wrap around again another histone core and go on to produce an architecture that looks like beads on a string. And those beads on a string, which are called nucleosomes, will then condense into a highly compact DNA protein complex called a chromosome. And there are 23 pairs of chromosomes in the human genome. And the chromosomes are cleverly numbered from 1 to 22. Those are called the autosomes, and the numbers correspond to the size of the chromosome, where the larger the chromosome, the smaller the number. So 1 is the largest chromosome, 22 is the smallest, and then of course there are the X and the Y chromosomes. And so every cell in our body has 23 pairs of chromosomes, one set from her mother, one set from her father. 22 autosomes, and then 2 sex chromosomes. Now, just for fun, this is what it looks like if you wrote out the entire genetic sequence of the human genome and take that human genome sequence and bind it into volumes. This is a picture of me at the Wellcome Museum in London, where you literally have the entire human genome on that bookcase, where each volume again, or several volumes, will correspond to the DNA sequence of the different chromosomes. Now, one of the things that was surprising about the human genome sequence when people first analyzed it is that they discovered that there are only 20,000 genes in the human genome. This was remarkable because people expected, at minimum, there would be 100,000 genes. But yet there was only 20,000 genes that make up the human genome, which meant that only 2% of the human genome consisted of DNA that coded for proteins, which are the workhorse molecules in the cell. The rest of the human genome looked as if it was junk. Most of the human genome looks like it's derived from retroviral infections. And there's other sequences in there of non-functional genes and what looked like evolutionary debris. And so people argued that the human genome is a wasteland of garbage. Just a relatively small percent is actually functional. And of course that begs the question, why would a creator produce a genome that looked like garbage? And so many people saw this as compelling evidence for biological evolution, but also saw this as a fulfillment of something known as the C-value paradox. Now, this is an idea that traces its origin back to the 1960s and 70s, when biochemists began to be able to quantify through laboratory techniques the amount of DNA found in individual cells. And so what they would do is they would isolate cells from organisms. Like human beings have 210 different cell types. They would isolate those different cell types, and they would measure the amount of DNA in every one of the cells. And what they discovered is that all the cells in the body of a particular organism has the same amount of DNA. This is called the C-value for being constant. It's a constant value. And so people thought at that time, well, the more complex an organism, the more DNA it's going to have in its genome. But when they plotted out the C-value as a function of organismal complexity, you get diagrams that look like this, which basically show no relationship whatsoever between the amount of DNA in the cell of an organism and its complexity. And the resolution to what's called the C-value paradox was effectively junk DNA. They argued, well, the difference is essentially due to junk DNA. It's just, again, accumulation of evolutionary waste. And so most of the genome has no bearing on our complexity. It's just, again, there as a vestige of an evolutionary history. And so the question then would be, not only why would a creator introduce garbage in the human genome, but why would there be shared sequences where the same sequence seems to show up in corresponding regions of the genomes of humans and the great apes? Well, the explanation for this would be that this junk DNA arose in the common ancestor, and as the lineages diverge, that junk DNA sequence would be carried throughout those lineages, and it reflects an evolutionary history. But interestingly enough, over the last decade or so, there have been some intriguing discoveries made that seem to indicate that junk DNA isn't necessarily junk, that we've been discovering in a piecemeal fashion that most of the classes of junk DNA actually play a functional role. And if that's the case, then of course you could say, well, if the junk DNA is functional, then the fact that it shows up in corresponding regions of the genomes reflects common design, not common descent. This was a concept we talked about last night. But in the midst of this, along comes something called the ENCODE project. This was a project that was initiated right after the human genome sequence was produced. The problem is you've got these bound volumes that have 3.2 billion genetic letters in them, and nobody knows how to read them. Okay, the best we could do is maybe fish out sequences for genes, but nobody knew how to read that sequence. What was needed is something like a Rosetta Stone. And so the idea of the ENCODE project was, it was an attempt to identify all the functional elements in the human genome so we could interpret the human genome sequence. ENCODE stands for the Encyclopedia of DNA Elements. And this project began in the early 2000s, and there was a pilot phase that cost $55 million, by the way. That's a pretty expensive pilot study. But these researchers took 1% of the human genome sequence and they tried to identify functional elements. And it turns out that they were successful doing that, so they inaugurated what's called phase 2 of the ENCODE project, where they scaled it up at the cost of about $130 million. And the phase 2 was completed in September of 2011, and we're currently in phase 3. Now, when this is all said and done, there's going to be about $300 million spent on the ENCODE project, which is a bargain, by the way, because it costs $3.2 billion to sequence the human genome. So for a tenth of the cost, we're going to be able to now interpret what the human genome is telling us. This was big science come to biology, where you had 440 scientists from 32 research groups around the world working on this project. They performed 1,650 experiments. They analyzed 147 cell types. There are 210 cell types in the human body, so phase 3 of the ENCODE project is filling in the missing cells. They produced 15 times 10 to the 12 bytes of data that required 300 years of computer time to analyze. When they reported the results, they published 40 papers that appeared in a wide range of scientific journals. And the idea here is that now that we have this data, we can finally make sense of human biology and even human uniqueness. We're going to be able to develop a better understanding of the genetic basis for diseases and maybe develop new ways to diagnose and to treat some of these diseases. But this insight also impacts the creation-evolution controversy in that the results of the ENCODE project completely undermine this whole case for junk DNA that buttresses biological evolution. Now, some of the functional elements that the researchers explored are listed here on this slide, and they all have to do with what's called gene expression. It's one thing to have genes within the genome. You can think of those genes as being like the words that are in a dictionary. But when you write a novel, let's say, you're going to use different words from the dictionary in different orders and in different frequencies and in different ways. And so, in a sense, the human genome is like the words in a dictionary, where it consists of essentially the set of genes that you need to build human beings. But then the gene expression changes from cell to cell, changes during the course of the cell cycle, changes during the course of development. And so each cell during different stages in the cell cycle, during different stages in development, represent different novels that have to be written. And this is all through gene expression. And it looks as if most of the human genome is actually involved in regulating and expressing the information in the genome so that it can be used to construct a human being. The researchers who reported these results basically argued that 80% of the human genome appears to consist of functional DNA. And in fact, they argued that as we move to phase 3 of the project, that it looks like it's going to actually move to almost 100% of the human genome. In other words, the human genome is not a wasteland of junk, it's all functional. It's all playing a critical role in making us human beings. And this has radically revised the way that we think of the human genome now. It's not a wasteland of junk, it's an elegantly designed system that's far more complex and sophisticated than anybody would have ever imagined. Now it's interesting, when I present this idea of the ENCODE project and how it's completely revised the way that we think of the human genome, the question I'm asked is, well, how do biologists respond to this? And something that the ENCODE project has done is precipitated something called the junk DNA wars, where you have two groups of scientists now in biology. One are the genomicists, who look at the ENCODE project as being valuable data, that they're using it to solve problems in biomedicine, that they basically accept the results of the ENCODE project and the idea that most of the human genome is functional. Then you have another group of scientists, primarily evolutionary biologists, who argue that the results of the ENCODE project cannot be correct, because if it is correct, it completely undermines key ideas that are part of the construct of evolutionary biology. And they basically argue that the scientists that did the work on the ENCODE project are incompetent, and that this is the worst piece of science that has ever been produced in human history. And it's something very interesting going on here. Those people that are ENCODE deniers are basically saying this, that the ENCODE project results cannot be correct, because if they're correct, it undermines the evolutionary paradigm. And so they're arguing, because of the primacy of the evolutionary paradigm, we know the ENCODE project results can't be correct. This is doing science backwards, because in science, the data determines whether a theory is valid, as opposed to the theory determining if the data is valid. And I literally had an engagement with P.Z. Myers, an evolutionary biologist, on Darwin Day this past year, where we were doing a debate, and we engaged on the ENCODE project, and P.Z. Myers, again to my face, said, it can't be right, because if it's right, then evolutionary biology can't be correct. And it's interesting, because the irony here is that this is not lost on actually genuinely objective scientists who are also interested in the ENCODE project. I'm going to close with a lengthy quote from a guy named John Maddox, who's a very well-known molecular biologist. And this is a statement that is coming from a scientific paper, where Maddox says this: there may be another factor motivating Grar and the related articles. These are critics of the ENCODE project, which is suggested by the sources and selection of quotations used at the beginning of the article, as well as the use of the phrase evolution-free gospel in the title. The argument of a largely non-functional genome is invoked by some evolutionary theorists in the debate against the proposition of intelligent design of life on Earth, particularly with respect to the origin of humanity. In essence, the argument posits that the presence of non-protein coding, or so-called junk DNA, that comprises greater than 90% of the human genome is evidence for the accumulation of evolutionary debris by blind Darwinian evolution, and argues against intelligent design, as an intelligent designer would presumably not fill the human genetic instruction set with meaningless information. This argument is threatened in the face of the growing functional indices of non-coding regions of the genome, with the latter reciprocally used in support of the notion of intelligent design, and to challenge the conception that natural selection accounts for the existence of complex organisms. This is mind-blowing, because again, John Maddox is not a friend necessarily to intelligent design, but he's actually calling out the ENCODE critics by saying that their motivation for challenging the ENCODE results has nothing to do with science at the end of the day. They're concerned that this actually provides evidence for intelligent design and undermines a key argument for biological evolution. And so I'm just going to wrap things up by just saying there is a new view that we have now of the human genome. And I presented this statement last night, but it's worth again presenting today. And this is a statement by Eric Green, who was the director of the Human Genome Research Institute at the NIH. He said, during the early debates about the Human Genome Project, researchers had predicted that only a few percent of the human genome sequence encoded proteins, the workhorses of the cell, and the rest was junk. We now know that this conclusion was wrong. ENCODE has revealed that most of the human genome is involved in the complex molecular choreography required for converting genetic information into living cells and tissues. I think that the human genome justifies a view that I would take and many other Christians would take, that human beings are the product of a divine creator, and that our genome is, and the nature of our genome is completely consistent with that idea and is no longer consistent with an evolutionary view of human origins. Thank you very much.